Good morning, everyone. Once again, thanks for joining me today. This talk is about the histology of the nervous tissue. The first lecture on this, and next week I will give you another class on this topic. What I would like to start with is today that I have to tell you some basics regarding this tissue. It's about basic tissues first, because this will be the final basic tissue you have to get familiar with. This is a new tissue. Until now in the lectures, you got familiar with the epithelial tissues already in the practices. You got that. Then the connective and supporting tissues came. Then the muscle tissue was also discussed in the lectures, and this will be the final basic tissue. This I would like to introduce a bit, because you are coming from very different countries, you are coming from very different schools, education systems. I have to introduce some basic terms on the nervous tissue. Some of you are already familiar with these terms, but some of you are not. And I have the experience in the exam that people are usually very well prepared for the minor details, but sometimes basic terms are somehow overseen. They are not learned properly. They are simply missing, and this is something that I really would like to avoid. So I will go through some basics of the basics regarding the nervous system, and then I will first turn to the nervous tissue proper. I will focus on the neurons today. I will classify them and tell you their main histological characteristics. And I will mention the glial cells too. And in the very end of the lecture, I will introduce the sensory ganglion and the autonomic ganglion, which are two slides. Also in the practice, you will meet those. Let's start with the recap of the basic tissues that you know. Where does this topic belong to? So epithelial tissues you are already familiar with. You studied those in the first 5 weeks in the practices, surface epithelia, glandular epithelia, and so on. You learned this already. Connective and supporting tissues you have studied also, with the collagen fibers, with the fibroblasts, with the cells, and so on. Muscle tissue is a topic that is discussed both in the lectures and in the classes, but this is the third type of basic tissues, and the final one is the nervous tissue. This is what I would like to start to introduce today. The basic terms, in order to understand what I'm talking about, if it's about nervous tissue, would be something that I want to go through. And as I mentioned already, some of you are already familiar with this. But first of all, the nervous system is something that has parts. And in this semester, we do not go into the details regarding the central nervous system, because that remains for the third semester that you study anatomy. That will be the neuroanatomy semester in a year for you. That is, of course, the brain and the spinal cord, and the topic is just a future issue. What I would like to talk about is more the peripheral nervous system, because if you study the histology, basically, if you get a slide almost from any of the tissues of the human body, you will eventually find parts of the peripheral nervous system in the slide. Therefore, we have to introduce the peripheral nervous system, which is made up by the nerves. The nerves, I will tell you in at histological level next week, but you have to know that these are fibers of bundles of nerve fibers traveling in the periphery, and the human body has 31 pairs of spinal nerves arising from the spinal cord, and we have 12 pairs of cranial nerves arising from the brain. The other thing what I would like to introduce today is the ganglia or ganglion or ganglia. These are associated to nerves, and some of them are indicated in this scheme also. Next to the spinal nerves, at their dorsal root, you have the sensory ganglia, for instance. Also, some of the sensory ganglia are related to cranial nerves. But we also have so-called autonomic ganglia, which are not indicated in this scheme. They will control a wide range of biological functions, and I will come back to this point later. So we will talk today about the peripheral nervous system. I will tell you less about the nerves, but more about the ganglia, and I will introduce the sensory and the autonomic ganglia today. So this was a kind of anatomical classification of the nervous system, but now we turn to a further classification of the nervous system. So how do we divide the nervous system into parts is the next question, and one can follow the function. So you have also a functional classification, and the functional classification is important for us because you will be medical doctors and you will study the functions, and you will see if anything goes wrong, you will realize that the function is lost. Therefore, you have to be clear. You have to be aware what functions does the nervous system have. And one part has the somatic. One part is the somatic nervous system, which has to do with your body in a way that it will perceive the signals from the environment. And this part of the environmental information will get into your mind and it will get conscious at the end. So it perceives stimuli from the environment, and those signals will be translated into electric ones, which then travel to the centers, to the highest order centers of the brain, to the central nervous system, and finally you will realize what happened in your environment, in your mind, so you get at the conscious level what happened. For example, if you make a coffee, you go to the coffee machine, you pick up the cup of the coffee, and then that mug or that cup is pretty hot. This is an information for your mind that if you grab that particular object which is hot, that may burn your skin, and this is something that you have to realize, because if you keep that particular hot object for a longer period of time, you may burn your skin, which means tissue damage. Or if you peel a potato and you accidentally cut your finger, you push the knife into your skin, that evokes immediately pain, which is perceived by the brain, and you know something happened, and you don't push the knife deeper into your skin because it may cause even a more serious injury. So this is the afferent or the sensory part, which informs you about the environmental challenges. Now you decide something to do. If you pick up the hot cup of coffee, you can decide what do you do. You may decide to put down on the table as fast as you can. If it's extremely hot, you may throw it away. You may smash it into the sink, that you don't get a deeper injury in your skin. So this is something related to the higher order decisions, which are made by the highest order centers of the brain. And the central nervous system makes a decision, and this decision is running again as an electric signal towards the periphery. These are commands, and you have to perform them, and this runs in an opposite direction from the highest order centers to the periphery, and then you will perform the movement as a motor command, and it goes in the efferent part of the somatic nervous system. In order to visualize this a bit, I prepared such a simplified scheme. So the yellow area is the environment, the white box would be the body, the green area is the central nervous system, and the dark yellow box wants to represent the peripheral nervous system. The environmental signal are called stimuli. It can be temperature, it can be the edge of the knife, it can be some taste information, it can be some light or some acoustic signals, some vibration, and so on. So many kind of stimuli come from the environment. And if it's the hot temperature, it might be the pain, or if the knife cuts the skin, it's again the pain. So it's a certain kind of mechanic stimulus, which then activates the receptors in your skin. I don't want to go into the details about the receptors because this belongs to the neurohistology, and I will give you a lecture on this next year. But the receptors are on the end, at the ends of the nerves, and certain kinds of nerve endings, and they translate the electric signals, the stimuli into electric signals. So the mechanical, chemical, and so on stimuli will be translated into electric ones. These are certain ion channels usually, and either through the spinal nerves or through the cranial nerves, the information goes to the highest order centers. So if you return to the example of the hot coffee, you pick up the mug, which is hot, you hold it with your fingers. They are innervated by the median and ulnar nerves, so they will carry the signal to the cervical spinal cord. If you put the cup of the coffee to your mouth and you try it, and then the hot coffee goes into your mouth, which is innervated by cranial nerves, and then the signal that the coffee is too hot goes through the cranial nerves straight to the brain. And then you can decide what you do. You put down the coffee, or you puke it out, or you swallow it. This is decided by the brain. And then after this afferent information way that from the periphery, the signal goes to highest order centers. Now the information spreads the other way around, and then you can perform something. You can drop the mug, or you can swallow the coffee, or you can puke it out if it's needed. So this is the signal that goes either through the cranial nerves or through the spinal nerves to the periphery to the effectors through the efferent or motor direction or way, and the effectors will perform those commands which are performed or which are made by the brain in order to drop the hot object or in order to swallow the coffee as fast as you can. So the effectors are usually responsible for that the signal of the nerve is transmitted to those structures which finally perform the command. This is usually muscles and eventually glands, because the movements are performed by mainly by skeletal muscles. If it's about the somatic nervous system, you decide if you stretch your fingers, then you use your extensor muscles, or if you grab an object, then you carry out a contraction in your flexor muscles. So this is how the signal will control the functions. And here I mentioned already the glands. And this is already related to the autonomic nervous system, because we do not have a voluntary control on the glands. If I would take a lemon now, and if I would eat the lemon here, and you would see that I'm eating a lemon, you would most likely have some more saliva in your mouth, because you see that should be very sour, and then you would also produce more saliva. But if I ask you to produce more saliva in your mouth, that does not really work, because we do not have a voluntary control on this function, as it is related to the autonomic nervous system. And the autonomic nervous system is autonomic because it's independent from our mind, from our decisions, since it works automatically, and the brain decides without a higher order voluntary control what do we want to do. This part of the nervous system has also an afferent part, which perceives again the information, but most of the times from internal organs this information comes, like the blood pressure, the stretching of the vascular walls, the chemical signals from the blood, like pH level, oxygen content of the blood, carbon dioxide content of the blood, or how much food you have at the moment in your bowels during your digestion. So one could give several examples, but this is something that we don't really know. So if I ask how is your blood pressure now, how much oxygen you have in your blood at the moment, this is something that you usually don't know, because it's part of the it belongs to the autonomic nervous system, and it does not reach the level of consciousness. But the brain does monitor continuously this information. And then depending on the environmental requirement or the internal requirement, if you have to adapt to your body to a certain situation, then the efferent part will perform what it has to do. So, for instance, if the blood pressure is going down and you have to increase the blood pressure, then the smooth muscles of the vascular wall will contract. If you have to produce more saliva because you have to start to eat because you are having your meal, then the saliva production will increase. Or in a stressful situation, for example, if you are driving your car and you come to a dangerous traffic situation that you have to brake unexpectedly because somebody is crossing the road, and unexpectedly you have to avoid that you crash. So this is something that is a stressful event, and then the body is adapted to these situations, like the heart rate is increased because the cardiac muscle will contract faster. This is controlled by the sympathetic nervous system, for example. Also, the exam situation is such a thing. So if you go into an oral exam, there are the cards, you have to choose one, and then you know that you didn't study a couple of cards, a couple of topics because you didn't have time for that. Then you have a higher heart rate because you are in a stressful situation at the moment. If you finally manage to pass the exam and you return home after the exam, because the last couple of days you couldn't eat properly anymore because you were stressed, then you are already relaxed, you open the fridge and you start to reload your energy storages. Then you have then more appetite. You can eat with a better appetite. And also, after having a nice meal, if you lay down on the sofa and it's Sunday afternoon, you are looking at the Formula One race, you sleep on the sofa, so you relax and you load up your energy storages. This is related to the parasympathetic nervous system, which is then more active at this moment, and the sympathetic one is inhibited. Also, the digestion in the bowels is controlled by the enteric nervous system. This is part of the autonomic nervous system, and this is a certain highly complex part of the autonomic part of the nervous system that would require alone a single lecture. In the third semester, when you talk about the autonomic nervous system, more detailed lectures will discuss this, but this is how we can divide the nervous system according to the function. And I think it was enough with the introduction. Now you know what the nervous system, what are the main parts of the nervous system, and you have now the nervous tissue, which is about histology. So the nervous tissue is, if we talk about the histology of that, is immediately divided into two main family of cells, or two main races of cells. The first, which has to come into your mind if it's about nervous tissue, is the neurons, the nerve cells, and these have the main function. This is what I will talk about more today. But don't forget that the other main family of cells is the group of the glial cells, and I will come back to those in a minute. But first, about the nerve cells, about the neurons. This is a statement which is quite important. Please learn it. Nerve cells are responsible and specialized for perception, transmission, processing, relaying of electric signals. So this sentence does not tell that much more than almost all cells can do, because cells can pick up signals. They can even sometimes transmit them. This is true for some muscle cells, some epithelial cells also. They may even process that signal and relay it, but what they are not, what is not characteristic for them is that they are not specialized for that. So this is the point here, that the nerve cells are specialized for the perception, transmission, processing, and relaying the electric signals. So this is their main function, and because of this, they are responsible for the control of biological functions, and they synchronize, they harmonize the functions of the human body in order to maintain the biological functions in a certain harmonic in a certain harmony, which means that if the nerve cells or the neurons or in general the nervous tissue is not working properly, that means that we cannot really control our biological functions properly. So some functions are lost, and in the worst case, then the harmony is completely lost, and because of this, we may even die. So therefore, the damage of the nervous tissue, the damage of the brain and brainstem, which contains the vital centers, will be not adjustable with life. So it is something that you have to understand, that the nervous tissue and the nerves will be responsible for the control, and if we lose the control, we lose our life. So this gives the significance of the nervous tissue also. And nerve cells are able to communicate with each other and also with other tissues, and this communication takes place through the synapses. I don't have time to talk about the types of synapses. I will give you a lecture on this in neurohistology in a year again. But the synapses are those places where the signal is going from neuron to neuron, or from the neuron to another type of cell. And as I mentioned, the nervous tissue has, besides the neurons, also glial cells. The glial cells were discovered and in the early era of histology and neurohistology, and people initially thought that these should be some cells which simply fill the gap between the neurons. They have possibly they thought they have some supportive functions, and that's it. Now we know that that this is far away from the truth, because the glial cells, besides their space filling and supporting function, which is true, the name glial also comes from the word glue in Greek. So they thought they should just keep the keep the nervous tissue together. So glial cells have, besides the supportive functions, a wide range of biological functions which will assist the functions of the neurons. They will feed them, they will pick up their metabolic waste products, they will break down neurotransmitters, they have immunological functions. So one could talk a lot about the glial cells, but because most of them are found in the central nervous system, I don't want to talk about that. That is also a future issue. But what I have to tell you is the significance of the peripheral glial cells. And in the periphery, two kinds of glial cells have a greater significance: Schwann cells and satellite cells. I will talk about these cells a bit later. Schwann cells next week, satellite cells at the end of this lecture will be introduced. Back to the neurons. If I want to classify the neurons, there are multiple options to classify them. The first option is that I take their function in consideration to classify them, and based on this, one can make a very simple classification. One can say we have neurons which have the function to pick up the signal. This is, the afferent part of the nervous system. This is the sensory neuron population, to carry the signal from the periphery to the central nervous system. Inside the central nervous system, we have neurons which allow the communication between nerve cells. They interconnect nerve cells with each other. Therefore, they are designated as interneurons. We don't talk about these in detail today. I will show you a bit later a scheme where you can see them, but it's not the main topic today. And I also mentioned that in the somatic nervous system, if I want to carry out something, a movement, then the information now spreads from the brain through the nerves into the periphery to the effectors. And this is performed by motor neurons, also called motoneurons or efferent neurons, and they will control the movements. And in the autonomic nervous system, they may also control the biological functions of internal organs, but I will come back to this point a bit later. Okay, so this was the functional classification of neurons. One can also classify the neurons according to their shape. according to the morphology. But in order to be able to perform that, then you have to know in general how a neuron looks like, and this is what I would like to explain in this drawing. So if I tell you nerve cell or neuron, most of the times people have in their mind something like a star-shaped cells with many processes, and this is usually also true for most of the neurons. They have a cell body, which is sometimes star-shaped, sometimes more round, but for sure it's called perikaryon or soma. And in the center of that, you have the nucleus, which has a well recognizable nucleolus if it's hit when you cut it. The cell body is this I mentioned. It's called in Greek either perikaryon or soma, or cell body in English. And from this cell body, most of the times multiple processes arise. These what I show you now are the dendrites. They have multiple branches. They may branch multiple times after each other. And their common feature is that they usually pick up the signal from other nerve cells. And also we have a longer process. One cell has only one such a process, and that is designated as the axon. And the axon has a typical origin, which is called axon hillock. And this is the spot where the electric signals of the cells are produced. This is the area where the action potentials are initiated. Next week, I will talk about this a bit more detailed. The axon will eventually branch. If that happens, the branching is typically perpendicular to the trunk of the axon, in contrast to the dendrites, which are sometimes branching also, but their branching is usually in a sharp angle, not in a perpendicular angle like axons branch. And finally, at the end of the axon or the axon collaterals, you have a so-called end tree or terminal arborization. Some books call it telodendrion. This is the official name, but end tree you may find in some books also. And as I mentioned, the dendrites may pick up the signal. They are responsible for taking up the electric signals from other nerve cells. Here you can see nerve endings coming from other neurons, and here you have synapses. And this neuron then will produce also electric signals, which runs through the axon, and finally it will be transmitted to another nerve cell, which is here, and this is another synapse. I don't want to talk about types of synapses and functions. This is a future issue, but now you see that how in general a nerve cell looks like. It has processes, dendrites, and an axon. But this is not always the case. We also have other kinds of neurons in shape, and therefore people designed classification for neurons according to their morphology. So there's a morphological classification of neurons to be studied, and this is this slide about. In very primitive animals, in creatures which are of a very simple nervous system, have these kind of neurons, so-called unipolar neurons, which do not have dendrites. This is the cell body, the soma, and they have one single axon, which may be branched at the end with an end tree, but this is a single axon. This is not very common in the human nervous system. In the retina of the eye, we have similar cells, but this is something that you will study later, not today. Also, in already a little bit or a little bit further developed, a bit more complex cell type is the bipolar neuron, which already has two processes. One of them receives the signal, that's the dendrite, and the other process is called axon. So two processes, therefore the name bipolar neuron. This is also not very common in the human nervous system. However, we do have it. In the retina of the eye, again, we have similar bipolar cells, and in the inner ear, there are some neurons which have this morphology. These are the ganglia related to the hearing and the balance. You will learn about this later. You don't have to know that much about these cells, but what you have to know is this kind of cell. And this is a funny thing, because this cell has one single process which splits into two branches, and the debate was always there in the literature. What to call these cells? How do we define them, or what terminology do we use for them? Because they have finally two processes, but these two processes come from one common trunk, and people invented the electron microscopy, and they started to study these processes, and it turned out that both have the morphology of an axon. So they don't have dendrites, and therefore it was hard to give a proper name. And finally, people designed this name pseudo unipolar, which means fake unipolar neuron. Why? Because it has one process. Because of this, it would be unipolar, but it goes into two directions. So finally, it is somewhere between the bipolar and the unipolar. But since the two processes are axons, it was difficult to classify. And nowadays, people usually use the term peripheral process or dendritic axon for one process. And the other one, which goes to the brain or to the spinal cord, is the axonic axon or central process. And similar cells you will find in the sensory ganglia, and this will be discussed today. Therefore, I will show you histological examples for that at the end of this lecture today. Okay, so this is the pseudo-unipolar neuron, which is characteristic for most of the sensory ganglia in the human brain. The cell has one process which splits into two branches, one dendritic axon, which histologically looks like an axon, but it has a sensory function, therefore it's called a dendritic axon, and the other process goes to the brain, which gives the signal further to the next cell. Because of this, it has the axonic function. Even if it's already an axon, histologically, we add the term axonic axon because it gives the signal further to the next cell. You may call these also central process. And the fourth type of neurons is that what I introduced in the earlier slide of mine. So a multipolar nerve cell with a soma, with processes, dendrites, usually you have many, but at least more than one. And then you have an axon, which goes usually to a far area. And for this, you can give as an example the cells in the central nervous system. The vast majority of the central neurons are of multipolar type, but because today I'm talking about the histology of the peripheral nervous system, I also have to give you an example for that. And the autonomic ganglion is the best example in the peripheral nervous system for the multipolar neurons. Okay, so this is the story of the classification of the neurons if we follow their morphology according to the processes and the number of the processes and types of the processes. So if I want to make it understandable once again how these cells work, I have to tell you something about the spinal cord. This is part of the central nervous system, and I started this lecture with that I don't talk about the central nervous system today. This is still true, but very briefly, in the spinal cord we have gray matter or gray substance and white matter or white substance, and that will be the gray matter will be the area where the nerve cells, the neurons, are found, and in the white substance or white matter, we have the nerve fibers traveling the pathways. We don't talk about those today. But let's go back to the original example. Again, I pick up my mug, my coffee, and then, or if I cut my finger when peeling the potato, then the pain is coming and it activates a receptor in my skin. And now a nerve cell will carry this pain as an electric signal through its peripheral process or dendritic axon, and then through its axonic axon into the spinal cord. If you look at this cell, this is morphologically a pseudo-unipolar neuron. Functionally, it's a sensory neuron. It is not alone because you have these nerve cell bodies in a group, And these are also pseudo-unipolar neurons which innervate different parts of the body, which are found in the same group of neurons. And this particular structure here, it's a group of neurons in the periphery. This is something that we call ganglion. And because functionally it contains sensory cells, you call it sensory ganglion, made up by pseudo-unipolar neurons. Please do not mix up the neuron term or the nerve cell with the ganglion. Why? Because the ganglion is a group of nerve cells, and within the ganglion you have multiple nerve cells. The information is, as I mentioned, going towards the higher order center, so it's a sensory or afferent information pathway. And the sensory ganglion cells are responsible for the signaling. And these are pseudo-unipolar nerve cells, and they form a group of pericardia outside the central nervous system, which means we are in a ganglion. Now the spinal cord knows that I have some pain in my skin, and through an interneuron most of the times, which connects neurons in the brain, the signal is going further through a synapse to another neuron, which is sitting here in the gray matter. This is called anterior horn of the gray matter. And from here, this neuron will give rise to an axon, which exits the spinal cord, and that travels in the periphery, and finally it will reach a muscle. And this muscle is innervated by this motor neuron. Now, the muscle can perform a movement. So if I picked up the hot object and I decide to drop it, then through this way I can cause a muscle contraction which stretches the fingers, and I drop this particular object. So this is a certain kind of reflex that the information, the environmental signal, activates the receptor, and the information goes through this cycle and will perform a movement at the end, and you react to the environmental challenge. So now you see what a sensory ganglion is and how this is interrelated with the somatic part of the central nervous system. And if I go on, one more thing I have to add. There is here a lateral horn of the spinal cord, and there are also neurons. They will have processes which go into the periphery also, but they do not reach directly the organ what they have to control, in contrast to the motor neurons here indicated by red. But they will stop in the periphery, and they will control another neuron which is in a ganglion. Therefore, this yellow indicated nerve fiber is preganglionic nerve fiber because it goes to the ganglion. That's why preganglionic. And then in the periphery, this axon ends and synapses with a neuron with a multipolar neuron in the periphery. Please compare. This was pseudo-unipolar. That is multipolar. So now with this multipolar neuron, it will communicate, and this multipolar neuron will be activated, and then this neuron with its postganglionic fiber, compare please the terminology, after postganglionic fiber, will innervate the organ what we have to control, and this could be an internal organ, for instance the heart. This one, what I indicated, is a simplified scheme of the autonomic nervous system in the periphery. It works with two neurons, with the preganglionic fiber that terminates and activates a second neuron that will then go with its postganglionic nerve fiber with its axon to the internal organ to increase the heart rate, for example, or to change the saliva production or gastric juice production, or I could give you several examples. Of course, again, this neuron is not alone, but you have multiple neurons in the same area. Again, I have a group of neurons outside the central nervous system. So what I'm talking about is again a ganglion. So this is again a ganglion, but now you have, however, you have a group of pericardia outside the nervous system, so it fits with the term ganglion, but please compare the shape of the neurons, the morphology. They are multipolar nerve cells, and they don't have the sensory function, but they have the function related to the autonomic nervous system to control internal organs. This is why we call this ganglion autonomic ganglion, because it controls independent functions, independent from our voluntary decisions, from our decisions, because this is related to the biological functions controlled by the autonomic nervous system. So sensory and autonomic ganglion. And before I show you the histological pictures of those, one more remark that I will come back to in the next week. Here I was talking all the time about processes of neurons going and coming through the periphery. They are not just randomly running everywhere. They are bundled into well-preformed connective tissue layers, sheets, and this bundle of nerve fibers in the periphery is called the peripheral nerve. This is what you must remember from your anatomy classes, that you have nerves which control muscles, for instance, or the skin. They provide sensory innervation as well. This is what I will discuss with you next week. But let's go back to the ganglia and let's have a look to their histological appearance. So we have nerve cells there. This is just a scheme of a neuron, what you can find in the Wikipedia also. I like this figure because many histological terms are indicated in this, so it's a kind of self-check before you go into the exam. If you read this and you can say some sentences about each term, you are kind of prepared. But how does this neuron look like in general? This is a high power magnification image of neurons in the spinal cord. They are multipolar cells with Nissl staining and with hematoxylin eosin staining. And please note that the cells are pretty blue with Nissl staining and pretty dark basophilic with hematoxylin eosin staining. This is related to their rough endoplasmic reticulum. The Nissl bodies belong to the rough endoplasmic reticulum. This is blue with Nissl staining, and it is basophilic with hematoxylin. The nucleolus is also well recognizable because that contains ribosomal RNA. You know from cell biology that the ribosomal RNA is produced in the nucleolus, and therefore it is pronounced. The nucleus of the cell with hematoxylin is euchromatic with some limited amount of heterochromatin. You know, the euchromatin is related to the high rate transcription of the DNA. These are metabolically active cells, therefore many ribosomes, and therefore euchromatic. All right, and how do these ganglia look like? They also contain these neurons. First, I will show you the sensory ganglion histologically. This is a middle range magnification of the sensory ganglion. What you have to focus on are these huge cells. They are 50 to 100, sometimes even 100 microns in diameter. So maybe not that big that as the fat cells, but comparable. They have a euchromatic nucleus with a well-pronounced nucleolus. And the cell is basophilic because of the rough endoplasmic reticulum in the cell. And because neurons are long lifespan cells, they will accumulate some aging pigment in their cytoplasm if the tissue sample is coming from an older person. You will see these brownish inclusions in the cells. That is the so-called lipofuscin pigment, and this is what you can find in long lifespan cells like in neurons. And also these cells are surrounded by glial cells, as I mentioned. So these were the neurons. Around them you have glial cells. Around the neurons proper, you have these so-called satellite cells. They support and they feed the neurons. They also remove their metabolic waste products. So they will support the neurons' functions not only mechanically but biochemically and in different aspects. Also, you have nerve fibers traveling here, and they have a sheath that is covered by Schwann cells. But this is something that I will tell you next week. This is not discussed today. So this is the hematoxylin stained preparation. But if I take the same section and I put it into silver nitrate solution, I completely, or for the first view, a completely different picture appears. Some stronger shrinkage of the cells will occur, but as a result, you can see the nerve fibers much more efficiently. You see here nerve fibers wrapping around the cell bodies, and each neuron has only one single process that loops around the cell body a couple of times, and then it splits into two branches. And this is how a pseudo-unipolar neuron looks like. So now you know that this is a sensory ganglion because you have these pseudo-unipolar processes. In the last slide, you couldn't see this because it was hematoxylin stained. But in this staining, it will be obvious that the neuron has one single process with a couple of loops, and it branches to a peripheral and a central process, as I explained you earlier today. You have also nerve fiber bundles passing here. The related cell bodies are in other locations, so you don't see those in this area, in this view, but these are nerve fibers here. And if you compare this black picture with the next one, which will be the autonomic ganglion, then you will expect there multipolar neurons and not pseudo-unipolar ones. And in this picture, now you see again black cells, but if you check one, for instance, if you pick this one, you see that it has basically in all possible directions multiple processes on the top, on the left, at the bottom, and also this is unfortunately impossible now because we are not sitting at the microscope. But if you would move the micrometer screw of the microscope and you would be able to move the table, you would see the third dimension also, and you would see that behind this plane and in front of this plane also in 3D like a star, in all directions dendrites arise from this cell body. And that demonstrates that here you are having a nerve cell that has many processes. So it's a multipolar neuron, and with many dendrites and with one axon, it should be in an autonomic ganglion because this has multipolar neurons and not the sensory one. Also, if you look at the picture, you can see smaller nuclei of cells impregnated. They are the satellite cells, again, supporting a nutritive functions you have to mention. And if you study this picture for a little bit longer, one can find very tiny processes also. They correspond to the axons. And here you can see again, just to remind you, we have the preganglionic fibers. They terminate and they synapse in these autonomic ganglia with the cells, with the multipolar neurons here. The synapse proper is hard to show. With this technique, it is not very possible, but there are some other ones one could use. But please memorize that in these ganglia, we have synapses. And in this synapse, we have, it's a chemical synapse, so there is neurotransmission by neurotransmitters taking place. And you have to know that the neurotransmitter in this ganglion is the acetylcholine. Okay, so these are cholinergic synapses. These neurons in the picture are controlled by the release of acetylcholine, which is a chemical neurotransmitter. Okay, I'm sorry about speaking 3 minutes longer. The time is over and I'm almost done. I'm done actually. If you would have any kind of questions, I'm listening. Otherwise, next week I will continue with the histology of the peripheral nerve. Thank you very much. Is there any kind of questions?
Sorry, Professor.
Yes.
In the autonomic ganglion, the first synapse inside the gray matter of the spinal cord. I want to know this preganglial fiber get the information from which nerve?
The autonomic nervous system is. I show you the slide again. It's easier to explain. Yeah, now you see. So you are talking about this neuron here that controls this neuron in the periphery, and here is the synapse. And your question is, what is this control? What is this neuron controlled by?
Yes,
Um,
exactly.
you have multiple sources. One is that from higher order brain centers, mainly from the hypothalamus, a nerve fiber pathway comes down from the brain through the white matter, and that will end on this neuron and it will control it. So if you see with your eyes that a very dangerous traffic situation happened, then your brain realizes that this is a life-threatening situation and you have to brake. And then this gives a signal to the hypothalamus, which is a brain part. And from the brain, from the hypothalamus, through this white matter, white substance, a descending nerve fiber bundle will end on this neuron. And that will activate it, which then increases the activity of this neuron, which will increase the heart rate. Okay, I highly simplify the story now, but later on, if we study the autonomic nervous system, you will get more details on this. But this is
And
pretty
can
much
we,
what happens.
and can we say instead of preganglial fiber, can we say it's efferent and afferent fiber, like somatic?
This one is efferent because it comes outside, but it's not
What about
somatic;
the
it's autonomic.
purple one? The
The
purple
purple
one?
one is also efferent because it is going to the periphery.
It doesn't have any afferent fiber.
There are afferent fibers also, but these are going on the backside into the spinal cord. It also has afferent parts, but I didn't want to indicate this because you would need the receptors. But in the cardiac wall here, you also have nerve fibers which will then arise in the heart, and in a similar way, like this, they would also enter, and they will be related to another neuron that is sitting in the sensory ganglion. You also have autonomic sensory fibers here.
Okay, thank you very much.
About the sensory part of the autonomic nervous system, you will hear a lot again in neuroanatomy, but this is for
It's
the
not for
basic
this semester.
histology. Yes, for the basic histology, this is not necessary because I just wanted to make understandable how what do these terms mean, and what does nerve fiber mean, and what does the cell body of the neuron do. And next week we will talk about the nerves. This is enough, and in the next semester, next semester, you will get deeper details on that.